Below you'll see the objectives for the Innate Immunity module. Today's is the fourth mini lecture of 5 covering this material. And this mini lecture will discuss the effector functions of innate immune cells. So we're going to go back to the local inflammatory response, and here we have a macrophage responding to all these bacteria. And as we've mentioned, this local inflammatory response induces the influx of neutrophils and monocytes from circulation within the tissue, where they can then migrate to the site of inflammation and help mediate clearance of this pathogen. So what are the key effector mechanisms whereby these cells do it? I'm going to divide this into 4 key effector mechanisms mediated by these inflammatory innate cells at sites of inflammation. The first is phagocytosis, that's mediated largely by macrophages, neutrophils, and eosinophils. The second is degranulation. Neutrophils are the most famous for this during most infections, but mast cells are also major contributors to this degranulation effect. And these inflammatory granules really ramp up the inflammatory response within the tissue. I'm also going to mention these extracellular DNA traps that are produced by neutrophils. And lastly, we'll talk about direct cytotoxicity, which is a function of natural killer cells. Before we do that, I do want to mention, macrophages are very complicated cell types. I mentioned that they are tissue resident. It turns out that they also have very specific functions depending on the tissue that they live in. And there are macrophage populations in the liver, the bone, the brain, and in many other tissues that have these specialized functions, such as Kupffer cells in the liver, osteoclasts in the bone, and microglia in the brain. We're not going to talk too much in host and defense about the function of these very specialized macrophages. It is as it goes beyond just functioning within the immune system. But one thing these macrophages all tend to share is their ability to participate in the process of phagocytosis. So macrophages express phagocytic receptors, and these receptors enhance the phagocytosis of pathogens. Now, macrophages are phagocytes regardless of whether these receptors are triggered or not. Macrophages are constantly sampling proteins and really any components of their extracellular environment. But phagocytic receptors can bind things like bacterial sugars and bacterial lipids, bacterial nucleic acids, and opsonins such as C3b and IgG. C-reactive protein is an opsonin as well. And when these phagocytic receptors are triggered, it allows these macrophages to take up these bacteria and shuttle them into an endosomal structure within the macrophage called the phagolysosome. The phagolysosome is a key structure for these macrophages and a key mediator of their effector functions. Here are some features that are important to know about the phagolysosome. First, the phagolysosome undergoes acidification. The pH is usually around 4 or lower. Within phagolysosomes occurs the process of oxidative bursts induced by the molecular complex NADPH oxidase. And this results in the production of superoxides and hydroxyl radicals and peroxides. There's also a process of nitrositive burst, which leads to the production of a free radical called nitric oxide that also has very potent antimicrobial effects. Within the phagolysosome, you also get enzymatic degradation. So we have lysozymes expressed. These induce bacterial cell wall degradation. Myeloperoxidase is expressed. This induces acidification and production of free radicals. Proteases, lipases, and nucleases are expressed that mediate destruction of the bacteria. These phagolysosomes are rich in defensins and other antimicrobial peptides. And one thing we'll learn as we discuss adaptive immunity later on is that phagolysosome function is enhanced in the presence of interferon gamma. This is a picture of NADPH oxidase on the right. It is a molecular complex composed of a number of subunits and catalyzes the production of superoxides and hydroxyl radicals. Now, there are mechanisms to also reverse this. And so macrophages express things like superoxide dismutase and catalase that reverses the effects of this oxidative burst. And these are important regulatory effects to prevent inflammatory responses, and especially phagolysosome responses, from getting out of control and killing the cell. What's interesting is that many bacteria have their own form of catalase, and it allows them to counteract the effects of oxidative burst and persist within the phagolysosome. And this includes Staphylococcus and Pseudomonas and Mycobacterium and so forth. A second key function of cells of the innate immune system is degranulation. And so as neutrophils, for example, become activated, and this can happen for mast cells, although the mechanisms of activation are slightly different, they produce and secrete granules. And these granules contain things like lysozyme, myeloperoxidase, these oxygen radicals, reactive oxygen species, and defensins, and then other inflammatory molecules that we'll learn a little bit more about later, called leukotrienes and histamines. And these are molecules that are highly vasoactive. They induce vasodilation. And so these granules induce kind of a little bit of tissue destruction, a little bit of vasodilation. It enhances the ability of innate immune cells to move around within those tissues and kill the bacteria. One thing to note is that these granules are very toxic, and the effects of oxidative burst are very toxic. And it turns out that most neutrophils that migrate into tissues at sites of inflammation and become activated there will die within 24 hours. And in fact, bacteria that induce a lot of pus, this pus is mostly dead neutrophils. A third mechanism that I want to mention is death by netosis. So neutrophils, as they die, can undergo a process where instead of undergoing apoptosis, they secrete their DNA and histones and form what are called these neutrophil extracellular traps. And they lose their chromatin structure, but this matrix is very sticky. It's rich in antimicrobial effectors like defensins, and so bacteria can get trapped here and be killed. Neutrophils die obviously in this process, but they're short-lived anyway within these tissues. And so this is one way that neutrophils can continue to trap and kill bacteria even after they've died. And then the last mechanism I want to mention is direct cytotoxicity by natural killer cells. As I mentioned, natural killer cells express both activating, indicated by the plus sign here, and inhibitory, indicated by the minus sign here, receptors that determine whether this natural killer cell is going to get activated and kill its target or ignore its target. Now, most cell types have lots of inhibitory receptors and few activating or inhibitory ligands and few activating ligands. And we mentioned that one of those inhibitory ligands in many cell types is MHC. And so NK cells that detect the presence of MHC and don't detect stress ligands that might be expressed by an infected cell will leave this cell alone. But infected cells often downregulate their MHC and increase the expression of these stress ligands, and this makes these cells susceptible to killing by natural killer cells. So as I mentioned, there's inhibitory and activating ligands, but when these cells decide to kill, they secrete granules and deliver them to their target. These granules contain two molecules, perforin and granzyme B. And when these when these are exposed to their target, perforin pokes holes in the membrane of this target cell. Perforin is actually highly related to C9. They're structurally homologous. It pokes holes in the membrane and allows granzyme B to enter into the cell and induce apoptosis of that target. This is going to be a similar mechanism that we'll learn about when we find out how cytotoxic T cells kill their targets. All right, so here are some of the key points of this lecture. Make sure that you review these key points to make sure that you've captured all the material that you need.